Hi folks, my name is Cole, and today on Investigate, Explore, Discover, we're going to be looking at tumor-infiltrating B cell characteristics in the context of breast cancer. Now, just to clear up, when I say cancer, I don't mean the best crabs around, so there's no need to get overly sensitive or hold a grudge about this, but this presentation is not about you. The cancer we're talking about today is a collection of diseases characterized by cells replicating uncontrollably. Now, cancer worldwide is fairly common, and it's likely that you know someone who has had some form of cancer. Worldwide, 20% of the population will develop some kind of cancer throughout their lifetime, which will prove fatal for 1 in 8 men and 1 in 11 women. Worldwide, the global cancer burden has actually been increasing, and increased to 19.3 million new cases and 10 million deaths in 2020. This puts it at the second leading cause of global death, and the numbers are almost double what they were in 2000. As you can see here by this map, cancer diagnosis seems to be concentrated in wealthier countries. And there are many different types of cancer, pretty much a cancer for every body type that you have. And for the first time this year, breast cancer is actually the most commonly diagnosed cancer throughout the world, beating out lung cancer. So to put that into context, there were an estimated 2.3 million new cases of cancer, which accounts for just over 10% of the new diagnosis in 2020 alone. Now, to know a bit more about breast cancer, we need to know about the breast anatomy. And I could have sworn that I got rid of all of these before, So breasts are made up of fatty tissue with lobules, which produce milk, which transport through ducts to the nipple. And there are many different types of cancer that affects these structures. However, today we're just going to be looking at invasive ductal cancer or carcinoma, which is the most commonly diagnosed form of breast cancer, and invasive lobular cancer or carcinoma, which accounts for about 10 to 15% of newly diagnosed breast cancer. In addition to there being many different types of breast cancer, there are also many different molecular subtypes that present similar characteristics. The ones that we're focusing on today, though, are the more difficult forms of breast cancer to treat, which express either human epidermal growth factor receptor 2, HER2, or are triple negative for markers. Our bodies actually have a system in place to get rid of cancerous cells when they initially see them. And that is our immune response. And depending on what type of cells localize to the cancer tissue, it will determine whether our bodies have a tumor suppressive response or a tumor progressive response. Now, as per the title, today we're talking about B cells. And B cells are in a unique position in that depending on their function in that particular tumor, they can tip the scales towards tumor suppression or tumor progression. B cells are an adaptive response cell and are critical in adaptive immunity. B cells commonly interact with T cells and prime their responses, but T cells and B cells together are termed lymphocytes. B cells themselves have many different functions. They can present molecular patterns in the form of antigen to T cells to prime their responses. They can produce antibodies, which are critical for humoral immune responses and are part of the reason why we have long-lasting immunity to infections and are also the reason why vaccines work. B cells also contribute to ectopic lymphoid follicle creation. And these are also known as tertiary lymphoid structures. Now, tertiary lymphoid structures are similar in structure to our secondary lymphoid organs, which are our lymph nodes and our spleens. Tertiary lymphoid structures, however, develop in non-lymphoid tissues, such as sites of chronic inflammation, which includes tumors. B cells can also produce cytokines that vary in nature, whether they be anti-inflammatory, such as IL-10, or pro-inflammatory, such as IL-6 or TNF-alpha. B cells themselves are actually a pretty complex subtype that have many different types of subdivisions. So upon exiting the bone marrow, B cells are termed naive and can travel throughout the body. But when they travel to our secondary lymphoid organs or tertiary lymphoid structures, they enter something known as a germinal center and are creatively called germinal center B cells. In the germinal center is where mature B cells proliferate, differentiate, and mutate their antibody genes during a normal immune response to infection. And upon differentiation, this spits out cells such as memory B cells, which can linger to repeatedly generate an accelerated and robust antibody-mediated immune response in the case of reinfection, and can also produce antibody-secreting cells in the form of plasmablasts, or plasma cells. So now that we know a little bit about breast cancer and B cells, this leads us to the most important question of every paper, which is, why is this important? So why is it important that we look at B cells in the context of breast cancer? This is important because in contrast to T cells, which are well characterized, tumor-infiltrating B cells are not well characterized. And understanding more about the role that these cells play can lead to better treatment options for those experiencing breast cancer. This leads us to the paper that we're looking at today, which is titled Tumor-Infiltrating B Cells Signal Functional Humoral Immune Responses in Breast Cancer by Garaud et al. Out of the Université Libre de Bruxelles in Brussels, Belgium. Now, I want to take a moment and say their title was really nice and actually gave me a bit of issue in coming up with my own title for this talk because this is ideal. But what we're going to do is we're going to jump right into the results that they found. And in this paper, the authors found that B cell infiltration is indicative of better long-term breast cancer results. And comparing people who had B cells present in their tumors to people who did not, there was a 20% better survival rate for both HER2 and triple-negative breast cancers. So the authors also looked at the immune cell populations in the breast cancer tissues and found that B cells can infiltrate these tumors. And as lymphocyte levels increase in the tumor, B cell levels also increase. Now, I mentioned that there are different subtypes of B cells, and the authors of this paper went and looked at these different subtypes in breast cancer. And they found that tumor-infiltrating B cell populations have low levels of naive B cells and high levels of memory B cells. They also found that there was an increase in germinal center B cells, and this indicates the possible presence of tertiary lymphoid structures. Additionally, they found that as germinal center B cells increased, there was an also correlative increase with T cell populations and an increase in antibody secreting cells. So this shows that humoral immune responses are generated at the tumor site. They also found that as germinal center B cell populations actually increased, there was an inverse relation with memory B cells and that those cells decreased. Overall, this indicates that tumors that have high levels of infiltrating lymphocytes and B cells are receptive sites for cooperative interactions between various active adaptive immune functions. The authors of this paper, in addition to looking at immune cells, also looked at tumor morphology and found that tertiary lymphoid structures are actually developing at these tumor sites. And so there's some really pretty microscopy in the paper if you want to check out exactly what that looks like. However, these tertiary lymphoid structures indicate that B cells contact other cell populations and again generate active humoral responses at the tumor site. To investigate B cells even further, the authors of this paper looked at the antibodies and cytokines that these cells secrete and found that tumor-infiltrating B cells release more antibodies and cytokines compared to B cells found in the periphery. And the cytokines that these cells produce prime T cells to a Th1 response and are producing a little bit more immunosuppressive cytokines than those in the periphery. So looking directly at the cells, the authors also found that tumor-infiltrating B cells have fewer antigen-presenting receptors. And this can be due to multiple reasons. It can be due to either B cell differentiating into plasmablasts. It can indicate the presence of exhausted tissue-like memory cells, or it can indicate a tissue microenvironment favoring immunosuppression. So they looked at these different situations and found that the decrease in antigen-presenting receptors are not due to differentiating, and also found that these B cells are reactive to stimuli. So summing this all up together, the authors of this paper found that as more lymphocytes infiltrate these tumors, and in particular B cells, there is a better outcome for HER2 positive and triple negative breast cancers. So this information is actually pretty interesting and contributes significantly to the breast cancer field. It contributes by characterizing tumor-infiltrating B cell populations and providing a better understanding of how these cells function in the tumor. By understanding this can lead then to more sophisticated cancer therapies for those experiencing breast cancer. This paper was really interesting, but there are always ways to take this science further. And so I had a few ideas of future directions of my own. First off, with the decrease in antigen presenting receptors, even though these B cells are able to stimulate themselves, are they able to co-stimulate T cells? Furthermore, because increased B cell populations are correlated with better cancer responses, is there a way that you can force translocation of B cells into these tumors? And along those same lines, tertiary lymphoid structures indicate humoral immune responses at the site of the tumors. So is there a way that we can induce tertiary lymphoid structure creation? Ultimately, though, these were just some of the ideas that I thought of. And what I'm also really interested in is what you think. What sort of ideas or questions popped into your head when hearing about this information? And is there any sort of topic you'd like to hear about in the future? So let me know about those in the comments below. Ultimately, though, I hope that you learned something and had fun doing it. So if you enjoyed this, give this video a like and subscribe for future content. And with that, I'll see you next time.